Assalamualaikum and hi, my name is Siti Suhaila binti Aripin. My final year project is on biodegradation of reactive green 19 by microorganism in digested sludge under anaerobic condition. Reactive dye is one of the examples of synthetic dye. Reactive dye is highly used in textile industry. The properties of reactive dye is highly soluble in water because it has strong affinity toward water. Reactive dye also consists of one or more azo and sulfonic group. This is the structure of reactive green 19. Digested sludge involves a process called sludge digestion, which organic solids are decomposed into stable substance. Microorganisms that usually found in digested sludge are Methanoceta conchyli and Methanoceta soingeni. The location of digested sludge is at the bottom of this tank. These are the parameters of digested sludge. It has total solid, volatile solid, chemical oxygen demand, pH and specific methanogenic activity. The characteristic of digested sludge is it has lumpy appearance, less solid, volatile content reduced about 50%, less objectionable odor, less active compound, and the color is brown-black. Problem statement is textile wastewater has carcinogenic property because it can damage our body system. It also high toxicity because it contains a mixture of chemicals such as toxic heavy metal, pentachlorophenol, and carcinogenic amines. The consequence is wastewater is released to nearby stream or river. It will lead to pollution because the water is contaminated with dye. When textile wastewater is released to river, it can cause harm to aquatic life such as phytoplankton, zooplankton, fish. The aquatic plant and phytoplankton cannot carry out the photosynthesis because sunlight cannot penetrate into the river. It also disturbs the water ecosystem. My objective of this study is to analyze the decolorization of reactive green 19 by microorganism in the digested sludge under anaerobic condition. My experimental design is first sample collection. Sample was collected at palm oil factory at certain Hilir in Negeri Sembilan. For the preparation of dye, the solution of dye was prepared for 1 gram per liter by adding distilled water. Biodegradation of reactive green 19 using digested sludge is performed in serum bottle. It is incubated for 7 days in incubator shaker under shaking condition. Analysis of degradation capacity is the data was analyzed using one-way ANOVA and 2-key Kramer multiple comparison test. For the result and discussion is this is the result for the decolorization capacity of reactive green 19 at 35 degrees Celsius. It showed that 300 ppm recorded the highest decolorization capacity. Same goes to the temperature 55 degrees Celsius. 300 also recorded the highest decolorization capacity. This is the comparison between the two temperature. As we can see here, both temperature recorded highest decolorization capacity at 300 ppm. Reduction of azo dye has been shown to occur under anaerobic condition, especially in association with anaerobic and facultative. Based on the results shown in Figure 1, 2 and 3, the yellow sludge had decolorized the reactive green 19 but with different decolorization capacity. Sample for reactive green 19 at 35 and 55 for 300 ppm show the highest decolorization capacity. The decolorization value increased as increases in dye concentration. The decolorization capacity of RG19 for 55 degrees Celsius is higher than 35 degrees Celsius. This suggested that the thermophilic condition offer increment in decolorization capacity within fixed time. For the concentration of 200 and 300 ppm in 55 degrees Celsius, there is no significant difference between these two samples. Since this experiment was carried out in shaky condition, under anaerobic, the degradation of azo dye still occur. Meanwhile, Pashuti et al. 2006 state there is no decolorization occur in shaking condition under aerobic. For the conclusion is reactive green 19 can be readily decolorized by microorganism in digestive sludge. The increase of dye concentration and temperature in anaerobic system has a significant effect on dye removal rate. For the future study, I hope to study the metabolism and pathway of microbe in digestive sludge degrading azo dye. Further optimization tests on carbon sources and nitrogen source inoculum concentration should be carried out. The parameters such as pH should be recorded in future study. This is all my reference that I used to complete this slide. Okay, that's all from me. Thank you. I hope you enjoyed this video.